In this video, we do colony PCR on our soil isolates to amplify the 16S ribosomal RNA genes. We will amplify the 16S rRNA genes from our 8 soil isolates, and we will use E. coli and Streptomyces venezuelae as positive controls and do a negative control with no template DNA. We prepare our templates by suspending a bacterial colony in 100 microliters of distilled water. 100 microliters of water is placed in each tube. A single bacterial colony is suspended in each tube of water using a toothpick. Twist the toothpick between thumb and forefinger to thoroughly resuspend the colony. Each soil isolate is suspended in a separate tube of distilled water, using a fresh, sterile toothpick each time. After suspending the last bacterial colony in water, all of the samples are heated at 95 degrees centigrade for 10 minutes. The heated bacteria are now ready to use as templates in our PCR reactions. We will make 2 separate master mixes for our PCR, one containing primers 27F and 1492R, and the other containing 27F and 519R. Each tube of master mix contains enough solution for 12 PCR reactions. We start by diluting our PCR primers in water. Our stock primers are at a concentration of 100 micromolar. We want them to be at 5 micromolar, a 20-fold dilution. We put 190 microliters of water into each of our tubes and add 10 microliters of each primer, making a total volume of 200 microliters. Note how we use filter tips throughout to prevent contamination of our PCR reactions. We are now ready to make 2 separate tubes of master mix. We add 60 microliters of the forward primer 27F to each of our master mix tubes. We then add 60 microliters of the 5 micromolar 519R reverse primer to one of our master mixes. And 60 microliters of the 1492R reverse primer to the other master mix. Each master mix gets 138 microliters of distilled water. We are then ready to add 300 microliters of the commercial 2 times TAC master mix to each of our tubes on ice. The commercial 2 times TAC master mix contains TAC polymerase, magnesium ions, buffer, and nucleotide triphosphates required for the PCR reaction. Finally, we add 30 microliters of dimethyl sulfoxide, DMSO, that I have been keeping thawed in my hand. Both master mixes are thoroughly vortexed and are then ready to put into PCR tubes. We put 49 microliters of master mix into each tube. Tubes with red labels contain the 1492R reverse primer. Tubes with blue labels contain the 519R reverse primer. When all of the PCR master mixes have been aliquoted out into the PCR tubes, we close all of the lids to prevent cross-contamination. We are then ready to add our templates. The first tubes are the negative control and contain no template. The next 2 tubes get the E. coli DS941 template. Each template goes into one PCR with 1492R and another PCR with 519R. All of the tubes now contain PCR mix and template, and we are ready to start our PCR reaction. The reactions start with an initial denaturation step for 5 minutes at 95 degrees. There are then 30 cycles of 95 degrees for 30 seconds, 48 degrees for 30 seconds, and 68 degrees for 2 minutes. The reaction finishes with a final elongation step of 68 degrees for 5 minutes. The tubes are inserted firmly into the heating block of the PCR machine. The lid is closed and the machine is started. When the heated lid reaches 104 degrees, the initial denaturation starts. The reaction is complete after about 2.5 hours. We will run the PCRs on an agarose gel on Thursday.